Ribosomes are nanomachines that make proteins in organisms, and their role is so vital for the cells is that they have to make their ribosomes rapidly and correctly. So most of the mutations that cause ribosome assembly defects result in embryonic lethality, while milder ones can cause neurodegenerative diseases, cancer, predisposition, or anemia. That's why we're interested in the Woolford lab in understanding how these ribosomes are assembled. Ribosomes are made of ribosomal proteins, ribosomal RNA, and 200 other assembly factors participate in this process. Importantly, ribosome assembly starts in the nucleolus, then moves to nucleoplasm, and it's completed in the cytoplasm where translation takes place. My project focuses on the remodeling events that occur in the nucleoplasmic stages of assembly. Our collaborations with research labs in China and Germany enable us to use a combination of approaches that include molecular genetics, proteomics, and structural biology. Recently, we found cryo-electron microscopy structure of assembling ribosomes during late nuclear stages of assembly. We found 3 structures that correspond to consecutive stages of ribosome assembly, and my project focuses on 2 of the remodeling events that occur in late nuclear stages. The first one is the rotation of a structure called central protuberance at the top of the ribosome, and the second one is the removal of what is called a food structure. By observing the electron microscopy structure, I am able to identify the critical interactions of RNA and protein that mediate these remodeling events. I study the molecular mechanisms that drive ribosome assembly by disturbing the molecular interactions of RNA and protein. I'm doing it in 2 ways. First of all, I'm looking at the protein composition of ribosomes and looking whether anything changed. And secondly, I'm looking at the structure of RNA by doing structure probing. This way, I added a lot of details to the remodeling events that occur before nuclear export to cytoplasm. And I found a new communication paradigm between the 2 remodeling events of ribosome assembly. Nuclear export is an important milestone in the journey of ribosomes as they move through cellular compartments, and it is the last licensing step before they reach their final destination in the cytoplasm, where they will start making proteins. This is why studying and understanding the mechanisms of ribosome assembly is very important, because these rearrangements stand as the quality control mechanisms to make sure ribosomes are properly structured to execute their function. Determination of the cryo-electron microscopy structures of ribosomes have been revolutionary for the field, and it definitely empowered the projects of other graduate students in the lab. Using these approaches, we will be able to understand how cells make ribosomes to make sure they provide proteins for their vital processes. All of the collaborations that we're doing within the department and outside of the department empowered my project tremendously, and I'm very happy to have that opportunity.